Yes. Hello.
Hello. Yes,
Yeah.
sir.
Yeah. Go on.
Can you make me a panelist?
Yeah, sure. Just a moment, huh?
Yes, sir.
Yeah, just a moment. Yeah. Okay, I have made it. Okay.
Yeah, thank you.
Okay.
Thank you, sir.
Yeah. Okay. Welcome. Okay. Acha, 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 acha. Okay. Oh, ho, ho, ho. Okay. I will send it to you today. Yeah. No problem. No problem. Okay. So once again, guys, last class. Okay. So last class, where did we stop? We stopped at your use of your microorganisms as your biopesticides, guys. Okay, so we had talked about your like different kinds of your bacillus, like bacillus thuringiensis, bacillus papillae, bacillus your lentimorbis. Okay, then we have fungi, that is your Beauveria bassiana. Okay, very important. Okay, Entomycorrhiza pallidus roseum and Zoothera radicals. Okay, and protozoans we have Nosema locustae, and viruses as your NPV nucleopolyhedrovirus or your granulovirus. Guys, these things they will be asked. Okay, so remember this thing, they will be asked in your exams, guys, either in fill in the blanks or name the following. So very important, you need to remember this thing. Okay, somehow or the other you have to go get this thing properly. Okay, but now let's try to move ahead, guys, and let's try to see the next thing, that is bioherbicides. Okay, so use of your microorganisms as bioherbicides. So, okay, let's try to see what really is going to happen out over here. So, first of all, guys, we need to know what do we really mean by a herbicide. So, herbicide is something that is going to kill a plant. Okay, so it is going to be responsible for killing your weeds, basically killing your plants, unwanted plants. So basically, whenever we talk about your weed, basically, or weedy side, okay, it is going to be like something which is like you have a farm. So let's say you are growing your monocots. Okay, and in those monocots, suddenly a dicot is going to come up. Okay, and that dicot is unwanted plant. So for you guys, that is going to be a weed. Okay, so always keep this in mind. So we are going to call it as your herbicide, or it can be your weedy side, guys. Okay. Okay, but now who are we really talking about, and what is really going to happen out over here? Okay, so we are going to make use of microorganisms as bioherbicides. Okay, so now guys, you are going to see that these bioherbicides or bioweedicides, they are going to be of two types. Okay, either you can use your fungi as your bioherbicide, or you can use your bacteria. Okay, so fungi or bacteria. Okay, but now guys, when we talk about fungi, you are going to see that these fungi, they are going to be of three types, and bacteria are also going to be of three types. So three types each. So who are we talking about, guys? Okay, so guys, in your fungi, okay, so when we are going to use fungi, guys, these are also known as mycoherbicides. Okay, mycoherbicides. Okay, and when we use bacteria, then they are going to be just known as your bacterial herbicides. Okay, so nothing special name as such. Okay, but now when we talk about these mycoherbicides, guys, remember we are talking about three. Now, who are we talking about? First one, we have Phytophthora palmyra. Very important. Okay, guys, Phytophthora palmyra. In your exams, guys, they will always ask you this one. This is the favorite one. Now, guys, what is this going to do? Now, how is this going to act as a mycoherbicide? So, guys, this Phytophthora palmyra, what is this going to do? Is this is going to control milkweed. Okay, it controls milkweed. Okay, now milkweed is basically a small plant kind of thing only in your different kinds of your orchards. So that way. Okay, so one moment. Yes, yes. After the class. Okay. So we have. Okay, 1 minute. Yeah. Okay. So once again we have this one. Okay. But then the second one. Now the second one, guys. Now this you need to keep the same in mind. Its name is Alternaria crassa. Okay, now same one, guys. Alternaria crosses, just like this. We have Alternaria neurospora. Okay, we have one more out over here, which is going to be used in your genetic experiments. Okay, so just keep this thing in mind, Alternaria. Okay, so now, guys, what is this going to be used again? This is going to be used against your water hyacinth. Okay, now water hyacinth, basically this is the one very very bad one. Okay, this water hyacinth, guys, this is going to eat away all the oxygen from the water. And if it utilizes all the oxygen from the water, guys, then what is going to happen, it is going to deplete the oxygen level. No oxygen is going to be available to all the other flora and fauna, and immediately it results into the death of the other organism. What do we call this thing, guys? We call this thing as eutrophication. Okay, so that is why this thing, water hyacinth, this is really really bad. But other important property of water hyacinth, what does this do? This is capable of, like, controlling, like, or absorbing minerals or like metals from that entire water. So that is one important characteristic of water hyacinth. Yes, Neha, go on. Yes, Neha. Neha, yes, you have raised your hand, Neha. You wish to ask something? Yes, it is ecornia. Fantastic. Yes. Neha, do you wish to ask something? Okay, seems like it was by mistake. Okay, guys, can you see the screen properly or is it blur? Quickly chat and answer. Okay, okay, fine. Okay, and then the third one, guys, out over here, the third one is going to be your Fusarium species. Okay, so we have Fusarium species, and that Fusarium species is going to be, like, working against most of the weeds. Okay, it is going to be useful against most weeds. Now, guys, how are you going to remember? Okay, you are going to remember microherbicides as PAF. Okay, keep this thing in mind. You just going to remember this as PAF, and bacterial herbicides, you are going to remember this as PAX. So PAF, PAX. Okay, guys, remember this thing. Okay, very very important. Otherwise, you will always confuse who is a bacterial herbicide and who is a mycoherbicide. Okay, but now when we talk about bacterial herbicides, who are we going to talk about? So the first one we are going to have is, guys, it is going to be your Pseudomonas. So we have Pseudomonas species. Guys, quickly tell me, earlier, where had we seen Pseudomonas? Pseudomonas and Clostridium. Quickly, guys, quickly. Where did we see it? Pseudomonas and Clostridium together. Guys, what were they known as? Anybody, quickly. Guys, they were your anaerobic bacteria. Okay, so keep that in mind. Okay, so we have this Pseudomonas. Guys, Pseudomonas, it's going to be against your weeds. It is going to be against many weeds. Same way, the second one, guys, the second one, we are going to have your Agrobacterium. Now, guys, this Agrobacterium tumefaciens. Okay, I believe the other man must have talked about this thing. So we have Agrobacterium tumefaciens in your, genetics and all. What we do is when we want to transfer some genes from an organism into a plant, we will always make use of this soil bacterium, Agrobacterium tumefaciens. Okay, so here we are just using Agrobacterium species. Okay, SPP meaning species actually, but anyways, I'll just put it down. Okay, so once again, guys, this is going to be against your several weeds. And the third one we are going to have is your Xanthomonas. Okay, so Xanthomonas species. So, guys, this is how you're going to remember this. This is you're going to remember this as your PAF and PAX. Okay, keep this thing in mind, guys. Very, very, very important. Okay, PAF and PAX. Okay, sure. But now, the next one thing that you are going to use, guys. Okay, so these were the normal ones like fungi and bacteria. But sometimes you can use your insects also as herbicides. Okay, insects as herbicides, meaning you are going to make use of these insects, like, and you're going to release them onto the plant so that they will eat only a particular type of a plant. And that is what is, guys, very important. So let's try to see the third one. Okay, now this is not in your the first time. We're going to see a little different thing. Okay, so something a little different. So insects as your herbicides. Okay, and now who are we talking about, guys? We are going to have two of them out over here. Okay, so the first one, guys, we are going to have something that is called as your Tyria moth. Okay, like I said, guys, see, you are using a moth. So basically, this is going to attack only one type of your specific plant. Okay, so what is this going to be used against, guys? This is going to be used against Senecio jacobic. Okay, so guys, remember this thing. Okay, it seems like a very different kind of a name, Senecio jacobic. Okay, Senecio jacobic, and the other mod that we are going to have, basically, it is going to be your Cactoblastis. Okay, Cactoblastis cactorum. Cactoblastis cactorum. And guys, without even knowing what this is going to work against, can you guess what will this work against, guys? Can you take a wild guess? Cactoblastis cactorum. Absolutely right, Sneha. Sneha Singh has given me the right answer. This is going to be against your cacti. Yes. Okay, so Cactoblastis cactorum. Okay, so guys, keep these two in mind. These are some new additions in your earlier versions, like in your like a couple of years ago. These were not there in your textbook, but now they have been added again. Okay, so this is what we need to know with regards to your entire bio herbicides, guys. Okay, everybody, all right with this thing? Okay, but now, guys, the next thing that we are going to talk about is how are we going to use microorganisms as biofertilizers? Okay, okay, so let's try to see. Okay, so now, now, guys, see, the main thing is, like, when we talk about your fertilizers, now, traditionally, which fertilizers were being used? It was your chemical fertilizers. But now, what happened? Over a period of time, we had its own problems. Like, those were all synthetic, they were all chemically manufactured. Then what happened? They were being used, like, unjudiciously, or they were, like, used, like, unreasonably. So what happened? Slowly, slowly, the pH of the soil altered, the kind of plants that grow on that soil, they altered. Plus, it is also responsible for killing all the good microorganisms. So there were many problems associated. Plus, there was leaching. So you could see, like, the fertilizers, they could leach through the soil and they could reach the underground water, and they contaminated the underground water. So whatever problems that we had talked about with biopesticides, guys, the same problems are with your biofertilizers. Okay, but now let's try to see, why are we going to use these biofertilizers? How are we going to go ahead with this thing? Okay, so we have our microbes as biofertilizers. Okay, But before we talk about these guys, okay, before we talk about these, let's try to talk about some normal fertilizers. Okay, so when we talk about these normal fertilizers, guys, what happens is fertilizers are divided into two types. Okay, so we are going to have inorganic fertilizers and we are going to have organic fertilizers. Now, traditionally, what are these? Let's try to see. Okay, so we have inorganic and we have organic. But guys, when we talk about the inorganic ones, what are these guys? These are the ones which are synthetic. They are produced artificially. And who are we talking about, guys? Okay, you are going to see that these are the ones which are going to include the NPK, nitrogen, phosphorus, and your potassium. Okay, so what happens is you are going to make like different proportions of this thing, and then you are going to be like spraying it onto the field, you're going to be dusting it onto the field. So those are the ones which are going to be your NPK fertilizers. Okay, but now the other ones, when we talk about your organic ones, so organic ones are what, guys? These are nothing but they are going to be obtained by your composting. Okay, by composting, or you must have heard of like khat or khad. Okay, so that is what this organic is like. You are going to take all your waste material, you're going to put it into a pit, then you're going to leave it for the microorganisms to act over it, and over a period of 3 months, then it is going to convert into some rich fertilizer, rich manure. That is nothing but your organic fertilizers. So what is this, guys? When we talk about this, you are going to have your farmyard manure. Yes, right. Okay, so farmyard manure, or you can also say that this is nothing but your compost or nothing but your green manure. Okay, so basically these are your normal fertilizers. Now, guys, whenever you use them, definitely the fertility of the soil is going to increase. But now, when we talk about your bacterial fertilizers, okay, or your biofertilizers as such, guys, what are they going to be? Since they are going to technically increase the fertility of the soil by adding extra nitrogen, or maybe they are going to add phosphates into the soil. So they will tackle something of that sort. Okay, so when we talk about, let's say, when we talk about biofertilizers, we are looking at your bacteria especially. Okay, so whether it is going to be your bacteria or whether it is going to be your blue green algae, cyanobacteria, or your blue green algae, what are they going to do, guys? They are going to add, okay, nitrogen, or it can be your phosphorus into the soil. Okay, etc, 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 we will see that. Okay, into the soil. And basically because of this, guys, we are going to have this nitrogen fixing bacteria. Okay, so especially if we have this entire category of nitrogen fixing bacteria. And guys, one of the most common examples is nothing but your rhizobium. Okay, so this is what we are going to talk about. So it can also include your blue green bacteria, it can be include your blue green algae, sometimes it is going to be your fungi as well. Fungi have a different role, we'll be talking about them, like in the case of your uh, ectomycorrhiza and endomycorrhiza, but that is a little later. Okay, so like this. But now let's try to see how it is going to be like, guys. Okay, so what really happens out over there? Okay, so generally when we have these biofertilizers, guys, what are we going to do? We take this bacteria and we are going to just either mix it in water so that the spores and all they get dispersed properly, and then we are going to just spray them all over the field. What happens? These particles, they are going to fall onto the ground. Wherever they reach the ground, immediately they are going to increase the fertility, because they are going to now grow, they are going to multiply and increase the fertility. Remember, these biofertilizers, they are going to be very very cheap. Okay, they are going to be cheap, they are going to be like eco-friendly. Okay, so they are going to be cheap, easily available, and they are going to be eco-friendly, so you don't have to really worry about polluting the environment. Okay, so this is what it is going to be. But now, guys, we have to talk about, like, how are these biofertilizers going to be like. So basically, biofertilizers, they can be of three different types. Okay, so either they can be nitrogen fixing, or they can be your phosphate solubilizing, or they are going to be compost making. Okay, then the other kind of category, they are going to be your cyanobacterial, because they are making use of cyanobacteria. And the third one is going to be your fungal biofertilizers. Okay, so they are going to be like, these three different types of your, the basic types, basically basic types of your microorganisms. Okay, so guys, let's try to see what really is going to happen out over here. How are we really going to see that? Okay, so first, just try to give you an idea. Okay, so we are talking about like the first one. Let's imagine we are talking about bacterials. This is going to be like, the nitrogen fixing one. These are the most commonly used, guys. So nitrogen fixing. Then the second one is phosphate solubilizing. Okay, so the phosphate solubilizing, guys, this is going to break down your phosphates and make these phosphates readily available into the soil. Okay, phosphate solubilizing. And the third one is going to be your compost. Okay. Second one in this category, guys, we are going to have your cyanobacterial. Now, these cyanobacteria, basically, they are going to show you association with different kinds of plants. Okay, so for example, we have your Anabina. Anabina is going to show you association with Anabina azole. Okay, and once again, what are they going to do with that association? Once again, they are going to bring about your nitrogen fixation. Okay. And now, guys, here also in this thing, in when we talk about nitrogen fixation, there are two types of nitrogen fixation. Either it is going to be symbiotic or it is going to be asymbiotic. Okay. So that is another category. And then from here, guys, we have the third one, that is going to be your fungi. Now, what happens in fungi, guys? There are two types of fungi. If the fungi, they are going to be present only on the upper side, meaning only on the outer side of the roots, then we call it as your Ectomycorrhiza. Okay, we are going to do this thing in detail, so nothing to worry. If they're going to be present only on the outer surface, then it is Ectomycorrhiza. And if this fungi, they penetrate into the root, guys, then that is going to be your Endomycorrhiza. Okay, so it is going to be this way. So Ectomycorrhiza and Endomycorrhiza. But now, guys, with this, let's try to see a few of them. The first ones, who are your nitrogen fixing ones? Okay, so one by one, we will start talking about all of these guys. Okay, so now the first ones that we are going to have are your, take a minute. Okay, guys, all right, fine, let's move ahead. Okay, chalo. Okay, so guys, let's try to start off with the first one, that is your nitrogen fixing bacteria. Okay, so let's understand. Okay, so in the case of your nitrogen fixing bacteria, guys, what are we going to see and what is the basis behind this? Let's try to understand this thing. Okay, so now, guys, when we talk about your plants, okay, so plants, let's say this is your tree. Okay, so whenever we have this atmospheric nitrogen, okay, atmospheric nitrogen, guys, there is 71% of your atmospheric nitrogen, which is going to be the largest. So technically, you will feel that there is absolutely no necessity, like of this nitrogen, or there is going to be no deficiency of this nitrogen out over there. Like you shouldn't be least worried about it, guys. The answer is no. Okay, the reason for this is this atmospheric nitrogen, this cannot be directly absorbed by the plant. It cannot be absorbed in this manner, guys. I need to convert this nitrogen into some form and add it to the soil, and then only it is going to be used up. So how does it happen, guys? So what happens is there are going to be these certain bacteria which are going to be present. Either they are going to be symbiotic or they are going to be your asymbiotic bacteria. What are they going to do? So what they do is, like, see, I cannot absorb direct atmospheric nitrogen. So these bacteria, they are going to absorb this atmospheric nitrogen, guys. And when they absorb the atmospheric nitrogen, they are going to convert it into your nitrites. Okay, they are going to convert it into first nitrites, and nitrites are then going to be converted into your nitrates. And now, as nitrates come up into the soil, these nitrates, they are now going to be taken up by the plant, like this. So you have to follow this entire process, guys. Okay, without this, the plants cannot take up nitrogen. Okay, and this entire conversion, guys, from your atmospheric nitrogen into your nitrates, this is done by your nitrogen fixing bacteria. Yes, if you want, you can see that entire thing. This atmospheric nitrogen is first going to be converted into ammonium compounds. Ammonium compounds are going to be converted into nitrites. Nitrites are then going to be converted into nitrates. Okay, so we are going to have nitrosococcus, and we have your nitrosomonas out over here, and we also have nitrobacter out over here. So that is that entire nitrogen cycle. Okay, but for the timing, it is not there for you. So I'm just giving you the brief up, like an idea about this thing. Okay, so guys, this thing is going to happen. So if you want, I will put down, okay, because someone has said. So first this is going to be like into your ammonium compounds. Basically, it is not exactly ammonia, ammonium compounds, then nitrites, and then nitrates. And nitrates are the only one which are going to be taken up by the plants, guys. Okay, always remember. So these nitrogen fixing bacteria, these are also going to be known as your diazotrophs. Okay, they are also known as diazotrophs. Okay, and guys, keep this thing in mind. In your exams, they ask you this thing. What are diazotrophs? Rather than calling this thing nitrogen fixing bacteria, they will use this sophisticated term. Suddenly, you will be like, "Oh my God, what is this? I don't know what is diazotrophs. It is nothing but nitrogen fixing bacteria." Okay, but now, guys, this nitrogen fixing bacteria, we can say that they can be of two types. See, I told you they are going to be of two types. Either they're going to show you association with the plant, or they are going to be free living. So let's try to see the next one, guys. So we are going to have symbiotic nitrogen fixers, and we are going to have asymbiotic nitrogen fixers. Okay, symbiotic and asymbiotic, or you can say these are going to be free living, or you can say these are also going to be your non-symbiotic. Okay, so either of the terms. But now, guys, first one, we are having this symbiotic ones, symbiotic nitrogen fixers. Now, who are we talking about, guys? Okay, so the first and the most common one, guys, okay, you are going to see them on the roots of your leguminous plants. Okay, so let's say this is your plant, something like this. Okay, and this is going to have your fibrous roots. Okay, now remember, these are your fibrous roots. Legumes are going to show you fibrous roots. Okay, can anybody tell me, guys? Okay, when we see this fibrous roots, does this plant have a taproot, or it is born with your fibrous roots only? Can anybody clear of this thing for me? Did it have a taproot, or is it just directly, like a separate type of root that is this thing? Okay, so only Komal has given me the right answer, and Sneha is also right. Okay, guys, now always remember, okay, always remember, whenever we have these kinds of plants, okay, earlier what had happened was this had a taproot system. Okay, it did have a taproot system, but then this taproot, it was short lived, it did not live long. So if it did not live long, it died. Now guys, if this one died, now what is going to happen? This is going to be removed. Now if the plant has to stay, if the plant has to stay alive, guys, it has to produce roots. Where are these roots going to come from? Now guys, you are going to see that, oh, this stem is going to have nodes and internodes, because that is a characteristic of a stem. So you will see now from the lower nodes, okay, now you will see some roots coming out. And what are these roots? These roots are nothing but your adventitious roots, or they are going to be known as fibrous roots. Now guys, you know why do we call them adventitious roots? Because it is accidental. Adventitious means accidental. Okay, so that is why, guys, this is going to be your adventitious root. And now because of this, what happens, guys, immediately now this plant starts surviving on its own. Okay, so this is happen. Yes, it is going to grow from any other part from the plant apart from the radical. Always remember, see, whenever we talk about the seed, okay, how is the seed? So you're going to see the seed, you're going to have the plumule, guys. Okay, the plumule and the radical. Here the radical is going to be short lived. Okay, the taproot is going to be short lived, and then it can grow from any other part of the plant apart from the radical. That is how your fibrous root system is going to be like. Okay, but now guys, what are we going to see? We are going to see that these roots, okay, now onto these you are going to see that, there are these nodules kind of thing growing onto this. Okay, now these nodules, how do they look like? They are going to look like some light brownish kind of, balls, like, full small chota chota gola gola sabudana kaisa hota hai, something of that sort, guys. You will see something of this sort. Okay, so we say that, these are going to show you symbiotic relationship. Okay, so now you are going to see nodules root nodules on leguminous plants. Okay, leguminous plants, guys, once again, who are we talking about? We are talking about your groundnuts, we are talking about your chana. Okay, these are all legumes. Okay, but now guys, let's try to see. I am having this root nodule. Let me just take this root nodule and let me just zoom in into the inside. Now, guys, the moment I zoom in into the inside, guys, you are going to see this, light pinkish kind of thing that you are going to see inside, guys. Okay, and what is this, guys? This is nothing but your rhizobium. This is going to be your rhizobium, guys. Okay, so this is very important. So guys, now different plants are going to have different types of rhizobium. Okay, so how's it going to be, guys? So we have, like, Rhizobium leguminosarum and Rhizobium physiole. Okay, so let's try to see. So we have two type of rhizobium, and they are going to attack different kinds of plants. So Rhizobium one leguminosarum and Rhizobium physiole. Okay, so guys, Rhizobium leguminosarum, this is generally seen in your pea, and physiole, this is generally going to affect your beans. Okay, so this is going to be your rhizobium. Okay, but now guys, just like we have rhizobium, is this the only one who's going to show you association? No, 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 no. It is not the one. Along with this, guys, we are going to have the other one, which is going to be your anabina. Okay, so we are going to have anabina. Now this is going to be your bacterium. Okay, this is going to be your blue green algae. So anabina. Okay, so anabina, this name is basically, this is anabina azola or azole, and this is going to show you symbiotic nitrogen fixation along with your Azola plant. Okay, Azola is a water plant basically, and it has some leaves. Okay, so Anabina is going to be perched below the leaf surface. Okay, so that way, and then we are going to have your Frankia. This is very important, guys. Okay, so Frankia, they will always ask you this. Now, where will they ask you in your NEET exam, guys? Identify who of them is going to be free living, who of them is going to be your like symbiotic this one symbiotic bacteria. Okay, so so guys, this is what we need to know with regards to your symbiotic nitrogen fixes. Okay, but now let's try to see the next one, guys. Okay, so the next one we are going to have free living. Now, as the name suggests, free living, yes, yes, right. व्हॉट इज बीन इन हिंदी और मराठी? बीन म्हणजे आपले काय बोलतो त्याला, किडनी बीन्स इज लाइक चवळी. Okay, you have different types of beans, basically broad bean, lima bean, black eyed beans, chawli. Okay, then we have rajma. Okay, so kidney beans is rajma basically. Okay, so basically it is an entire category. Okay, so now guys, let's try to see the free living ones. Now the free living ones, guys, as I'm saying that they are not going to show you association with any plant. They are directly going to be like free floating into the soil. But now, who are we talking about out over here, guys? Very, very, very important ones. Okay, so first one you are going to have azotobacter, very important one. Azotobacter. Second one, we are going to have nostoc. Azotobacter, we are going to have nostoc. Then we are going to have your Klebsiella pneumoniae. Okay, very, very important, guys. Guys, these examples are very important. Then we are going to have Clostridium. See, Clostridium and Pseudomonas, so that Clostridium. Guys, do you know like Clostridium, any disease caused by this Clostridium? Anybody? Anybody? By any chance? Food poisoning. Very nice. Okay, one is food poisoning and the other one is going to be your tetany. Okay, so Clostridium botulinum is going to be responsible for that food poisoning. It gives you the most toxic poison, botulinum toxin. Guys, if you have ever heard of that Botox treatment, that Botox treatment is because of this thing only. Okay, the Clostridium botulinum toxin. Botulinum toxin is Botox. Okay, and the second one, guys, it is your Clostridium tetani. So this Clostridium tetani, guys, it is going to be free living into the soil. Okay, its spores, they are generally going to be found on your rusted objects, rusted metals, or they're going to be on your old glass, or it can be on your old patara kind of a thing. Okay, and then what happens is, if you by any chance get a cut, the Clostridium spores, they are going to be lodged deep inside your wound. Now, what happens, guys? The moment your spores enter the deeper layers of the skin, okay, and into the muscle, inside the atmosphere is not going to be aerobic. Okay, keep this thing in mind, guys. Inside the atmosphere is not aerobic, it is going to be completely anaerobic kinds. Why? Because we are not going to see that our body is not going to be filled with hawa types. Okay, understand this thing. So now, the moment it goes into your muscles, you are going to get warmth, that is essential. You are going to have moisture, essential. Plus, you're going to have anaerobic kind of conditions, essential. Immediately, this Clostridium starts multiplying. And what does it produce? It produces your tetanus toxin. And guys, that tetanus toxin is going to be really, really deadly. It is going to bring about your muscle spasms, and then you are going to just contract. Okay, and then you're going to be like, am I. It is going to be that way. Okay, so really, really deadly stuff. But yes, this is going to fix nitrogen for you guys. Okay, the another one that we are going to have is your Bijerinca. Okay, given by a Bijering. Okay, Bigerinkia. Okay, sure. So etc. 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 So these are some of the important ones who are going to be free living or non-symbiotic. Okay, guys, just quickly have a look. Just see if you are okay with this, and then we can move ahead with the next part. Beigerinkia. So it was given by Beigerink. Okay, so Beigerrinkia. Okay, so guys, this was the first category. Let's try to move on to the next one now. Okay, so the next one, you are going to have your phosphate solubilizing ones. Okay, now whenever we talk about this phosphate, guys, okay, phosphate is going to be really, really important, or phosphorus as such is going to be very, very important for our body. Can anybody tell me where is this phosphate going to be utilized, guys? Phosphate utilized in our body? There are several places, but one place which is going to be the most important one, guys, where phosphate is going to be used. No, 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 no. Not in process, but some part. Some important part where it is going to be used. That is going to make the basis of your entire body. Yes, Devang has given me one of the right answers. It is going to be ATP. That is going to include that phosphate. But then something more important than ATP. Yes, Neha Singh has given me the right answer. It is going to be your DNA, guys. So, guys, once again, when we talk about your DNA, how is it going to be? You are going to see that, oh, you are going to have the pentose sugar. So, you are going to see the phosphate, which is going to be attached to the pentose sugar, guys. See this? It is going to be pentose sugar, and then this is going to be attached to your nitrogen base. This is nothing but your phosphate, guys, and we need to have this phosphate. But then how is this phosphate? Is it going to be like readily available? No, 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 this is not readily available, guys. So what happens is I need to break down this insoluble, okay, organic phosphate or inorganic phosphate, and then I have to create it into soluble phosphate, or some phosphate compounds. So once again we are going to require these bacteria. The bacteria, they are going to break down insoluble inorganic phosphates, and they convert it into your soluble, or they are going to convert it into your normal, which can be phosphate, which can be taken in, or phosphate, like your phosphate compounds, like sodium phosphates or your potassium phosphates, something like this, and then they are going to be taken up by the plants. Okay, but now, guys, who are the ones? Who are these phosphate solubilizing bacteria? So we are talking about now, guys. See, we are going to have your bacillus. Okay, first one, bacillus polymyxa. Okay, now, guys, these names are a little different. Okay, but you have to remember this thing. So bacillus polymyxa. Okay, we have micrococcus. So micrococcus, bacillus polymyxa. Then we are going to have your agrobacterium species, basically agrobacterium species. Okay, we are also going to have your aspergillus. Okay, and then once again you are going to have your pseudomonas. Okay, so pseudomonas species. Okay. Now, guys, how I like to remember this thing, I like to remember this as AMBAP. Okay. AMBAP. If you want, you can remember it this way. You will not forget. Okay. So this is nothing but your phosphate solubilizing bacteria. But now the next one we are going to talk about is, guys, we are going to have your compost making ones. Okay. Chalo, so let's try to see how are your compost ones really going to be like, guys. Okay. Okay, so now, guys, when we talk about your compost, okay, so what is really going to happen out over here? Let's try to see. So let's say we are having all your organic, inorganic waste, whatever it is. Now it can be your, like, it can be your dung. Okay, it can be your urine. It can be your kitchen wastes. Okay, it can be your kitchen waste. Okay, it can be your leftover food. Okay, it can be your paper. Okay, etc, etc, etc. Now what are you going to do in compost is, guys, you take this thing and you are going to dig a pit into the ground. So let's say you have taken a pit like this. Okay, now you take this entire thing and you are going to fill it. Now how do you fill it, guys? You are going to actually mix it with your water. Okay, all these things you're going to mix it with water, and then you are going to have a layer of waste like this. Okay, so we have waste. Then on top of that waste, we are going to have a layer of your soil. Okay, this soil is going to contain your dung and a little bit of your urine soil. And now you keep on repeating this thing. So layer of waste, layer of soil, layer of waste, layer of soil, layer of waste, layer of soil. And this thing keeps on happening till we reach the top. Okay, so let's say we have almost reached the top out over here like this. Okay, and now you are going to keep it out over here like this. Okay, so this is your last soil. Okay, the last thing you're going to like keep it like really, really nicely. So this is going to be your soil. Okay, now what are you going to do, guys? Now you are going to see that you leave this thing as it is, okay, for 3 months. Okay, roughly about 3 months you are going to keep it this way. Now in some cases what people do is, people will mix this thing up. Okay, in normal traditional one we were not mixing this thing. Okay, some people like to just turn this upside down, but this is not needed. Now what is going to happen, see, our compost is not yet prepared. Now after your 3 months into this pit, guys, now what are we going to do? We are going to take this and now we are going to arrange it in your conical hips. So you're going to take this material, you place it in your conical hips like this. Okay, and then you are going to, let's say this is, I'm just showing only one conical hip, guys, like this. Okay, and now once again you are going to cover it with your soil. Okay, so let's say now I'm covering it with soil. Now this thing, guys, you have to keep it. Okay, you have to keep it for minimum 2 months. So this is going to lay like this for 2 months. But then after this 2 months, guys, now what is going to happen? You are going to see that whatever bacteria, fungi, everything that was there present inside, those are going to convert this. Okay, convert this entire waste into a black powdery substance. Okay, this would convert into black powdery substance. And now what is this black powdery substance, guys? We call this thing as your humus. And this humus, guys, this is going to be really, really rich. Okay, this is going to be really, really nutrient rich. So now once we have got this humus, guys, you can take this thing, you can sprinkle it or add it to your water, and then add it to your soil, and then that's it. That is going to be your natural fertilizers. Now who all are going to be like working out over here? See, there are many bacteria, many things out over here. So I will just randomly write it out over here. So we are going to have bacteria, we are going to have your fungi, we are going to have some very important bacteria which is called as your acinetobacters, okay, or your acinetobacter, okay. And then we are also going to have some protozoans who are going to come out over there, and rotifers are like, small worm like structures. Rotifers, sorry, protozoans, sorry, first protozoans and rotifers, all of them guys, okay, they are all going to be out over here. Okay, once again, after soil part, okay, so once again, what is going to happen when you add this soil, you're going to keep it like this for 3 months, then you're going to convert it into conical hips and you're going to cover them with soil. Okay, so this is going to be your soil on the top, and then you're going to leave it just like that for 2 months under the sun. After 2 months, what is going to happen, you're going to see that that entire material is converted into black powdery substance which is called as humus. And humus is going to be like really really rich, nutrient rich, so you can just take this, add it to your soil, and it increases the fertility of the soil. Okay, fine. And these are all the bacteria. But now guys, this compost process guys, it is going to be a very very long process. Okay, people don't have patience for this, and that is why they will go for your like chemical fertilizers and all. Okay, also guys, remember there are different varieties of this compost. Now I am not using earthworms out over here. Had I used earthworms, then what is this going to be known as? This is going to be known as vermicompost. Okay, but then for the time being, I'm not using earthworms out over here. Okay, then if you are making this especially out of your dung, cow dung, okay, cow dung or cattle dung, then we call this thing as shenkhat. Okay, so if anybody is into gardening and all, you know this thing, like if you go to even a nursery, okay, you will always find that those people have those shenkhat packets out over there. Yes, manure. Okay, so we call it as, I mean, they label it as manure, but it is actually your shade cut. Okay, so guys, this is what we need to know with regards to your compost making bacteria. Everybody all right with this? Just have a look, guys. Okay, right. Okay, guys, what we will do is we'll just break over here for like 5 minutes. Anybody who wishes to go and have water, use the loo, go and come quickly. Okay, in 5 minutes, once again, we'll start with the next part, guys. Okay, chalo. So guys, once again, let's start. Is everybody back? Okay, guys, chalo. So let's try to move ahead and let's try to see a few more things, guys. 1 minute. Yes. Chalo. Okay, so guys, let's move ahead. Okay, and let's try to see the next part. Okay, so the next part we are going to have is your cyanobacteria. How are these cyanobacteria going to be important? Okay. Now, so guys, once again, when we talk about your cyanobacteria, there can be two different types. Once again, they can be free living, or they can be symbiotic, or they can be like aerobic, or they can be anaerobic, not anaerobic, but aerobic only, or they can be your photosynthetic or non-photosynthetic ones. Okay, so there are many varieties out over here. Okay, so let's try to just quickly put them down, like, how are they going to be, like, what happens. So cyanobacteria, they can either be your this thing that is, they can be free living, or they can be your symbiotic. Okay, then these can be either showing you nodules, nodules meaning nodule formations out over there, or they may not be nodule formations. Okay, or not. Okay, or these are going to have some special structures called as your heterocysts. So you will be seeing heterocysts, or they may not be producing any kind of heterocyst at all. Okay, so these are also going to be something. Now, guys, when nitrogen fixation takes place in those who have heterocysts, this is where nitrogen fixation occurs in your heterocyst. That way. Okay, but now, guys, when we're talking about these, like, what are the basic examples, guys? Okay, so we are going to have the like, if you're talking about the non-symbiotic ones, okay, and the symbiotic ones, some of them can be asymbiotic, some of them can be symbiotic. Okay, so basic example, when we talk about, we are having your nostoc, we are going to have nostoc, anabina. Guys, always remember the spelling of anabina is anabina. Okay, but the pronunciation is different. Okay, the next one we have is your lectonema, lectonema and oscillatoria, etc., 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 guys. Okay, so these are basically your cyanobacteria. But now, guys, out of these cyanobacteria, we are going to have tan. Okay, now how do I remember this thing, guys? I remember this as open. Okay, I remember this as opan. Okay, this way. So you have oscillatoria, plectonema, anabina, and nostoc. And from here, guys, if you look at the next one, that is your tan. Okay, we have tan, that is going to be Tolipotrix. Okay, Tolipotrix. Then we are going to have your Olocera and nostoc. Okay, or we have your Tolipotrix, anabina, nostoc. Okay, Tolipotrix, anabina, and nostoc. You will see that these are also going to show you some association with your lichens. Okay, so lichens, and some of them with your azola. So some of them, guys, if you look at these, they can show association with some lichens. And now, if we look at only anabina, guys, anabina can also show you association with your azola plant. Okay, so we have anabina azole, and guys, it is also going to show you association with cycas. Now, can you tell me, guys, if you remember this thing? See, I'm sure you have done this thing. In cycas, cycas is going to show you some special kind of roots. where you're going to have these nodules onto the roots. Can you guys tell me what are these roots called? The nodulous roots of cycas. Anybody, anybody. Very nice. Sneha has given me the right answer. Very good, Sneha. Very nice. Sneha Singh. Okay, those are your coralloid roots, guys. Okay, so it helps in the formation of coralloid roots. Very good. Very nice, guys. Okay, so this is what is really going to happen, guys. Okay, so that is what we need to know with regards to your cyanobacteria. Okay, now guys, what are we doing? First, we are just doing the classes of your organisms. Then we have to study few bacteria. Like we have to talk about, like, how is your rhizobium going to be like? How is your acetobacter going to be like? Azotobacter going to be like? So we have to talk about those organisms as well. Okay, and finally, guys, the last one we have said that we are going to have your fungal we are going to have your fungal biofertilizers. And these fungal biofertilizers, these are nothing but your mycorrhiza. Okay, now why do I call this thing as mycorrhiza, guys? Because they are going to show you association with your roots. Okay, so what do I mean by this? See, myco, whenever I use the term myco meaning fungus, and rhiza is nothing but your roots. So these are the ones which are going to be associated with your roots. Now, who was the first person who actually spoke of them? The first person to speak of them, his name was Frank. Okay. And now what did he say? He said that, oh, these mycorrhiza, we can classify them into two different types. That is ectomycorrhiza and endomycorrhiza, depending upon how they are going to work. Okay, so we have first one, ectomycorrhiza, because we'll talk about this first. This is simple. And then we'll move on to your endomycorrhiza. Okay, so now first, guys, let's try to understand what exactly is going to happen out over here. So let's imagine, guys, we are going to have a root out over here. Okay, now when we talk about this fungi, guys, now what do these fungi do? They are going to form an entire mycelium coat. You will see that they will form an entire mycelium coat onto the upper surface of your roots, like this, guys, completely onto this thing, meaning it is like, just like, forming a socket, or it is forming a sock like structure on top of the root, guys. But now what is so important, why is this going to help the plant? Guys, this fungus, it is not going to cause any infection or something, but rather what is going to happen, see, these mycelia, they are going to act like your root hair, and they are going to start absorbing whatever moisture, whatever water, and minerals and all from the soil. And then later on, it is going to make it available for this entire plant, guys. This is what is going to be important. Everybody getting this thing, guys? Sure. So this is what, your ectomycorrhiza is going to be like. Okay, so always remember, first of all, this fungal mycelium. Okay, so I just mentioned out over here, the fungal mycelium creates a mantle or a jacket on top of the roots. Okay, and the second important thing is, because they are going to do this thing, guys, they are going to absorb large amounts. Okay, absorb large amounts of water and nutrients. And guys, if you're talking about nutrients, especially, we are talking about NPK, that is nitrogen, phosphorus, potassium, calcium, etc. Okay, so this is what is going to happen. Okay, and now sometimes what happens is, okay, now guys, this is something very different. Okay, sometimes what happens is, only rarely, okay, sometimes they may penetrate. Okay, mostly they are going to be like, on the outer side only. Sometimes few of the mycelia, they penetrate into the root. Generally this is not a characteristic, but they may penetrate into the root. And what they give rise to is they form something called as Hartig net structure. Okay, they form something called as Hartig net structure into the roots. Basically, the Hartig net into the root cortex, guys. Okay, so this is what is going to happen. So one matter, guys, let me just quickly pull out a picture of this Hartig net so you'll get an idea how it really happens. Just a second. So, guys, always remember whenever we talk about this, in the case of your drought regions, in the case of your, where there is a scarcity of water, scarcity of minerals, okay, or if the atmosphere is like, really, really dry, in those cases, these are going to act like, really, really important, guys. Okay, so they will be like, they will definitely help the plant to grow in much better manner. Okay, so this is one. But then after that, the next one that we are going to have, guys, that is even more better. That is your endomycorrhiza, because that is actually literally going to enter into the roots. So one second, guys. So let me just show it to you, this Harting net structure. Yeah. Okay, so guys, first of all, see, this is how it is going to look like, your ectomycorrhiza. And see, what happens is, you are going to see that, see, out over here, okay, it is going to stay on the top only. So when it stays on the top, it is going to form that sheet kind of thing. So that is your mantle. Okay, but I told you, like, from here, not all, okay, not all, only some of them, they are going to enter inside. And when they enter inside, they are going to form this structure, like, a net like structure, netting kind of a thing. That is called as your Hartig net structure, guys. Okay, so here what we are seeing is nothing but Hartig net structure, but I will show you in one more better manner how is that going to happen, guys. Okay, so that you will see it. See, guys, see, if you look at this structure, you can see some of them, they have gone inside and they have formed a network kind of a thing. That network is nothing but your Hartig net. This is what is going to happen, guys. Okay, everybody all right with this thing? Okay, but now let's try to move ahead, guys, and let's try to see the next thing. Okay, the next thing is going to be your en- endomycorrhiza. Okay, and in endomycorrhiza, guys, you are going to see something different happening. Okay, so let's try to see what exactly is going to happen in your endomycorrhiza. Okay, so guys, first of all, here what is going to happen is, once again, exactly on the same lines. Okay, but now here, instead of, like, you are going to see that, instead of this mantle staying on the top, guys, what you are going to see is, oh, these fungi, mycelium, they are actually going to enter, they penetrate into the plant. Okay, and now if they penetrate into the plant, guys, there are two things. They can actually penetrate into your xylem also, and sometimes they can penetrate into your phloem. So basically, what happens is, these two things, the fungi and the plant, they are growing as a single entity kind of thing. Okay, because now both of their things are like completely joined. But now what happens? So then what is the difference? Now here, when they are going to penetrate inside, guys, what are they going to give rise to? They are going to show you these small, like, nodes, nodules kind of thing. These nodules kind of a thing, basically they are called as your arbuscles. And that is why, because they produce this arbuscles, guys, we call these as your vesicular arbuscular mycorrhizae, or we also call them as your WAM plants. Okay, so let me just put this thing down. So this is going to be your vesicles and arbuscles. Okay, and they will produce this empty space which is your vesicle. Okay, let me just go down out over here and let me just show you a little picture who's going to give you a comparative idea how this is going to look like. Say, I told you this is going to be that vesicle kind of a thing that is going to be formed, and you will see these arbuscles, these small nodes kinds of thing, guys. That is how it is going to look like. In the next case, okay, if I have got one more picture which I had downloaded for you guys. So let me just put it down out over here. Okay, so if you look at the difference, okay, let's see how the difference is going to be like. So if you look at your endomycorrhiza, guys, you are going to see that, see, the endomycorrhiza, this is going to go inside and it generally helps in forming these small balls like structures. That is your arbuscles. Okay, whereas, guys, you look over here, this is going to be your ectomycorrhiza. The mostly you're going to see that the mantle is going to be on the top side, and few of this mycelia are only entering inside. This is the biggest difference, guys. Biggest difference. Okay, everybody getting this part properly, guys? Understanding this? Sure, no issues. Okay, so this is like the biggest difference between your ectomycorrhiza and your endomycorrhiza, guys. Okay, so let me just tell you, and here we are not seeing it, but guys, but then okay, I will show you the this thing as well. So the vesicle as well. So let me just insert either. So it will form like small circular, like this, like and besides this it will form some circular kind of thing which is going to be known as your vesicle. I will draw it out over here so that will be better. Okay, so something of this sort out over here, so it is going to result into this. So vesicles. Okay, so guys, let's try to put this thing down. Let's try to see what happens. So here, what is going to happen? I told you these are also going to be known as your WAM plants. WAM plants or vesicular arbuscular mycorrhizae. Vesicular arbuscular mycorrhizae or WAM plants. Okay, but then what happens in this thing, guys? The most important thing, the mycelium, they penetrate into the cortical cells purposely. Okay, so they are going to penetrate into the cortical cells, and here we are going to see that they are going to absorb huge amounts of minerals and your water, just like how it happened in your ectomycorrhiza. Okay, and guys, if we have a comparison, like if we are thinking about, like who is better from these two, like ectomycorrhiza and endomycorrhiza, always remember endomycorrhiza, this is going to be more fruitful, this is going to be more better as compared to your ectomycorrhiza. And also remember, guys, when you use this endomycorrhiza, okay, in any field, it converts your less productive field into your more productive field, into more productive field. And guys, this is very important. productive field. Also remember guys, if by any chance there is a drought area, or if there is like an area where you don't have proper amounts of nutrients, okay, deficient soil and all, the use of these WAM plants, okay, is going to be more important. And why? Because these will make sure that the plant will survive. So that is really important regarding this WAM plants. Okay, everybody all right with this thing? So this is the difference between your Ectomycorrhiza and Endomycorrhiza. Guys, just have a look at this thing, see if you are okay with this. Then we'll move ahead with the last part of the chapter, like what are the different types of bacteria that we are going to use. Okay, the screen is blurred. All right, I will have to zoom in so that you will be able to see only these two. Okay, guys, very important thing if you want. Okay, so here this Hartig net. Okay, they will always ask you what is this Hartig net structure. Okay, and here they will ask you what is this vesicular and vesicles and arbustles. Okay, and sometimes, okay, in one year, in one of your board papers, guys, what had happened was they had asked differentiate between ectomycorrhiza and endomycorrhiza, and this was asked for like 2 marks. So guys, if you have 2 marks, then at least a minimum of 4 points you have to write down. Okay, how it happens is in your HSC, guys, every point is for half mark. In your ICSC, guys, it is 1.1 mark. Okay, but here you have to write 2 points for a single mark, guys. Okay, everybody all right with this? Okay, quickly guys, quickly chat yes or no. If you're okay, then we'll move ahead. Yes, okay. Sir, is it like 1 minute? क्या है ये? Sir, is it like in HSC we have to write full statement and fill in the blanks? Yes, you have to write full statements. Yes. Okay. See, those who are from HSC, those who are from SSC, guys, for you it will not be any problem, okay, to write the HSC board papers, because it is exactly the same thing. It is all going to be your subjective. Okay, you have to write. But those who are from ICSC, guys, when you are going to shift from ICSC to HSC, see, the entire idea is going to be different. In ICSE it was more on like you write 1 point, you get one answer. Okay, it was more of the objective type questions. But no, here it is going to be subjective. The more you write, the more marks you're going to get. It is that way. Or at least if you are going to write it to the point properly, then you're going to get proper marks. It is that way. So guys, writing practice is a must. Now what we have always observed is like most of the kids, even if they know all the answers properly, they have like by hearted everything, like word to word ditto, full ratta. But then what happens at the time of writing? Then suddenly their speed cannot match. So whatever they are like thinking, they cannot write it down, they cannot put it down, and eventually what happens? Suddenly, the last part of the paper, they have just lost it. It's that way. So that should not happen with you guys. Okay, so remember, writing practice is equally important. See, knowing everything is not an art, but to finish the paper in the stipulated time, that is your art. So to put down what is like important, who are the important points, that is going to be very like crucial for you. Okay, sure. So guys, let's try to move ahead. Let's try to see the next thing. Now see, we had talked about like these bacteria. Okay, like who are the bacteria that are going to be used as biofertilizers? So we spoke of, like we have your Rhizobium, we have your Azotobacter, we talked about your Anabina. Okay, so who are these going to be like? What are they really going to look like? So that is what we are going to finally put down. Okay, just a second, guys. Okay, so the first one out over here, guys, we are going to talk about the most famous, that is nothing but your Rhizobium. Okay, so let me just show you how the Rhizobium is going to be like, what really happens out over here. So, okay, so now let me just show you a few pictures, guys, how it is going to be like and what happens. Okay, so just a second. No, this, not this, not this one. Ah, okay. So guys, when we talk about your Rhizobium, okay, so I told you it is going to form these root nodules. So how is it going to happen, guys? See, this is how it is going to look like. So these are like, small sabudana kind of thing. That is nothing but your root nodules. But now, guys, when we isolate these, okay, when we isolate them and we look into your microscope, I mean, when we look into, we grow into the plate, okay, how it appears is it is going to look like this. Okay, and now when we talk about the microscopic image, okay, so in your laboratory it is going to grow like this. And now when we take a microscopic image, guys, we are going to see that, oh, this is going to look something of this sort. Okay, it is going to appear somewhat pinkish, because in gram staining it appears to be somewhat pinkish. That is what the thing is. Okay, so this is how it is going to look like. But now let's try to see, guys, what really happens. What do we really need to know, and what are the important things that are present inside of it? So, guys, there are going to be, like, two important genes which are going to be present in Rhizobium. One is the Nod gene and one is the Nif gene. They are the ones who are going to help in, in making this thing survive. Okay, and 1 minute, there's one more structure there which I wanted to show you. So see, guys, if we took take a section of this root nodules, okay, so I had shown you one section earlier as well. So this is how it is going to look like, guys. See, completely filled with your, the outer one is the root nodule, and inside we are going to have the Rhizobium, that way. Okay, sure. So guys, with this basic information, let's try to see how is it going to be like, what really happens. Okay, so guys, basically when we talk about these, they are going to be, first, they are going to be a rod shaped. Okay, so they are going to be bacilli like. In fact, when we talk about them, they are going to be irregularly shaped, like, they look like, X and Y structures kind of thing in your microscope. Okay, so first of all, these are going to be rod shaped. Second thing, these are going to be motile, guys. Okay, they are movable. Once again, they are going to be aerobic, but they are going to be gram negative. Now, guys, what this is gram negative and gram positive? Now, whenever we have, like, like staining of your microorganisms, bacteria especially, okay, we are going to see that bacteria pick up either two colors. Now, we have an entire staining process, okay, which is called as gram staining. So those who are going to appear pink, they are going to be gram negative, and those who appear to be violet, they are going to be gram positive. Okay, so we say they are going to be gram positive. This is the basic difference. Okay, basic difference between bacteria. But now, how are they going to be, guys? They are also going to be nitrogen fixing. So convert atmospheric nitrogen into your nitrates. And finally, I told you there are going to be two important genes. we have the nod gene, and we have the nif genes. Now, what are these genes responsible for? Nod is nothing but your nodulation. So it is the nodules. This is responsible for nodules. And nif gene, this is going to be responsible for nitrogen fixation. Okay. Another important thing, guys, because we said this is going to be root nodules, what is this going to show you? This is going to show you association with leguminous plants. Okay, so it is going to show you association with leguminous plants, and then it is going to have the root nodules, and these root nodules inside them, it is going to grow like luxuriantly. Now remember, inside the root nodules, this is also going to produce something called as leg hemoglobin. Now, what happens, can my question to you guys is, have you guys done leg hemoglobin anywhere previous year? I'll give you a little bit of idea. Okay, inside the root nodule, you don't want oxygen at all. Okay, you don't want free oxygen because then it will hamper the process of nitrogen fixation. So what does this thing do? Okay, the plant and this, the rhizobium, both of them, they are going to start producing leg hemoglobin. What does this leg hemoglobin do? Yes, absolutely right. The leg hemoglobin is now going to take away all the oxygen. Now, if the oxygen is removed, now the nitrogenous enzyme is going to be, as it is. Because otherwise, if there was oxygen, the nitrogenous enzyme cannot work. Now, if the oxygen is removed, now the nitrogenous enzyme is free to work, and it is immediately going to convert your atmospheric nitrogen into your nitrates. That is the job, guys. Okay, so keep that in mind. I'm sure someone has answered over here. So Sneha has answered. So I'm sure this must have been taken. Okay, so that's it. And guys, once again, we have said that we have two types of rhizobium. So we have your rhizobium leguminosarum, which is going to be for your bee, which is going to be for your peas, and we have your rhizobium phaseole, which is going to be for your beans. That's it. That is what we need to know with regards to your first one. Okay, but now the second one, guys, we are going to talk about your azotobacter. Okay, so let's try to see how is this azotobacter, what is really going to happen out over here. So guys, once again, let me just pull out a picture of azotobacter. Okay, so guys, when we talk about azotobacter, see in your this thing. So basically, this is going to be a free living organism. Okay, and when this is free living, guys, when we cultivate it into your labs, this is how it is going to give rise. It is going to produce like this mucus kind of colonies. It is very ropey kind of colonies. Let me just show you one more image how it is going to appear to be. See, it is going to look something of this sort. Okay, like very gummy kind of things, stating that it is going to have a polysaccharide coat kind of thing. Okay, so this is how it is going to be like. But now, when we zoom in into it and when we see this thing, guys, how is it going to appear? So you are going to see that this azotobacter, this is going to appear somewhat this purple kind that we have said. But see, this is how it will actually look in your microscope. Actually, it will look like this, guys. Okay, sure. So once again, with this basic information, let's try to move ahead, guys. How is this going to be? So first of all, this is going to be free living nitrogen fixing bacteria. Okay, once again, this is going to be nitrogen fixing. Okay, third thing, this is going to be your aerobic. Okay, non photosynthetic. Okay, here, is this going to form any nodules? No, this is non nodule forming. Okay, and plus this is your asymbiotic. So basically this is your free living, so it is a symbiotic one. Okay, but now even though it is non-nodule forming, guys, you will always find it like, closely associated or closely present. Okay, it is not going to be forming in nodules, but it is going to be closely associated with the roots of your grasses. Okay, but now, guys, when we are going to use this thing, okay, when we are going to use this as fertilizers, where are we going to use this thing for? We are going to use it for the cultivation of rice, cotton, okay, etc., etc., etc. All the rice varieties, guys. All the rice varieties we can use this thing for. Okay, chalo. So this is going to be your azotobacter. Now, just like we have azotobacter, guys, now we are going to move on to your azospirillum. Okay, azospirillum is also going to be your free living. So actually, azotobacter, azospirillum, they are in the same category, free living nitrogen fixers, guys. So this is in spelling error. Okay, azospirillum. Okay, but now once again, like I said, okay, first of all, let me just show you how does this azospirillum look like. One minute, guys, let me just take a quick image. Okay, so guys, as the name suggests, okay, azospirillum. Okay, so azospirillum meaning it has to be spirillum, spirillum meaning it has to be some spirals. So this is how it is going to look like. See, guys, it is going to be like small spiral structures, or we have a huge image out over here that is, but then that image is not going to be distinctive enough to show you that. uh, Azospirillum, no, 1 minute. Let me just quickly search if I can get one more image of Azospirillum. Yeah, okay. Okay, so guys, see, this is like another one which we have got. So see, this is how it is going to be, Azospirillum. Okay, so once again, very important nitrogen fixing bacteria. Let's try to put down, guys, how it is going to happen. What really do we need to know out over here? Okay, so first of all, like I said, guys, this is going to be free living. Okay, just it comes in the same category as your azotobacter, guys. Okay, so once again, this is going to be free living, a symbiotic. Okay, this is going to be aerobic, once again, nitrogen fixing bacteria. Okay, but now, guys, what is this going to be associated with? Now, this is not going to be forming any root nodules kind of a thing, but closely associated with the roots of your jawar. Okay, jawar, wheat, corn. Okay, so this thing. Now, guys, you know what? The most important thing about this is, guys, this is going to fix huge amounts of nitrogen. Okay, so high amounts of nitrogen fixation. And if we're talking about nitrogen fixation, guys, this is going to be around 20 to 40 kilograms. Okay, in an entire year, guys, this is huge amounts. Okay, that is what we need to know about azospirillum. Okay, but now the next one we are going to have anabina. Now, guys, we've already said we have here ana no, anabina, anabina azola, or your azole, and your other anabina. Okay, so let's try to see how is this anabina. Let me just give you an idea. Okay, so guys, basically, if we look at anabina, you are going to see it to be something of this thing. Okay, now guys, what is this? This is going to be your blue green algae. So this is going to have, like, small, these heterocyst kind of thing. And let me just give you an idea what really is that heterocyst. Heterocyst is the place where you are going to see actual, this nitrogen fixation taking place. Okay, so this is how your anabina is going to be like. Okay, and let me just take me like take one more example. Okay, yeah. So I will show you. Yeah, one more thing. Okay, so see, this is how your anapina is actually going to be like. Okay, but okay, let's try to see what do we really need to know. Okay, so first of all, guys, this is a blue-green algae. How is this? You can see that this is going to be a completely multicellular. Okay, so this is multicellular, and this is going to be somewhat filamentous. Okay, then what is this going to do, guys? This is going to show you nitrogen fixation. That is why this is important. Next thing, where exactly is this nitrogen fixation going to take place, guys? This nitrogen fixation takes place in these rounded bodies. These rounded bodies, guys, or these structures, these are special structures. These are called as heterocysts. So nitrogen fixation, this takes place in your special rounded structures called as heterocysts. And now, once again, guys, we said there are going to be two types. Okay, so we said, okay, so this is going to show you association with your cycus. And in the case of cycus, it is going to show you this cororoid roots. And in the case of your this thing, okay, your anabina zola, okay, this is going to show you the, like, the basically it is going to affect the leaves that way. Okay, and now because we are talking about anabina azola, guys, we are going to talk about this azola plant as the next one, the last one. Now what happens in your azola plant, let me just quickly show it to you. Okay, so guys, when we talk about your azola plant, it actually looks like this. Okay, it is a small plant with bilobed leaves kind of thing. Okay, let me show you a even better image. So it is like, small bilobed leaves kind of thing. Okay, so see, it is something of this sort, bilobed leaves all over. Okay, but now what happens, guys, when we talk about this anabina? Okay, you are going to see that this anabina has leaves, and inside that leaves there are going to be some small empty spaces. Those empty spaces, those are called as your erenchyma. Okay, so let me just show you on the dorsal side, guys. This empty space, it is called as your erenchyma. And that erenchyma, it is now going to be infected, or it is going to be like, it is going to show you anabina azola present inside of it. Okay, so there is one image like this, and now I'm going to show you another image where anabina azola is present inside of it, guys. So if you can see this, okay, so this is the same erenchyma thing, and then it is going to be present inside. Okay, so let's try to see with this basic information what do we need to know. Okay, so first of all, when we talk about this azola plant, okay, so azola plant, it is going to have bilobed leaves. Okay, it has this bilobed leaves, and these bilobed leaves, you're going to see that we have ventral lobe, which is going to be on the top side, and we have going to have the dorsal lobe, which is going to be on the bottom side. But now this dorsal lobe, guys, okay, now remember, this dorsal lobe, this is the one which is going to be infected. Okay, so you're going to see that inside this dorsal lobe, you are going to see this erenchyma. Now, guys, why are we seeing erenchyma? This erenchyma is nothing but your parenchyma cells only. But what are they storing? They are storing air inside of it. Why do I need this thing? So that it is going to give buoyancy to the entire plant, so that the plant does not fall. I mean, it does not sink into the water, and it keeps on floating. Now we are going to see that, oh, this erenchyma, now you're going to see that, oh, inside of this, now you're going to see your anabina being purged inside this anabina. And now what is it going to do? Anabina is going to bring about nitrogen fixation inside of it that way. And that's it. That is what we need to know with regards to your anabina. Okay, so this is the last one that we needed to know. Okay, so guys, once again, is everybody okay with all these organisms that we have done, guys? Everybody okay with this? Sure, just have a look at this, guys. Have a look at this. And then we have a very small part that is like, what is the use of your biofertilizers, which is like very, very easy, nothing to be worried about. Now, guys, earlier, okay, in your earlier editions, like, a couple of years ago, there was no separate mention about all these organisms. They had just spoken of this thing. But since last year, they have updated the textbook, and now you have all these organisms coming out over there. Okay, so remember, you need to know them because they might ask you a question, right? A short note on the different types of microorganisms used as biofertilizers. Then in that case, you will have to write, with respect to, like, two or three organisms minimum. Okay, and finally, guys, let's try to see what are the uses of biofertilizers. Very, very simple, nothing to really worry. The benefits. Okay, so first thing, this is going to be, it is going to be like a really, really cheap. Okay, so not at all expensive. You just take the bacteria, just apply it into your places, and then it is just going to start multiplying on its own. Okay, second thing, it is going to be eco-friendly. Doesn't harm your environment, doesn't leach into anything, nothing at all. Ultimate aim, we are going to increase the fertility. So increases soil fertility without, you know uh, without changing the pH or adversely affecting the pH of your soil. So increases soil fertility. Along with this, guys, along with, all this, like, like nitrogen fixation and all, sometimes, other compounds may also be added. Okay, so other compounds can be added into this thing, are added into soil. Now these can be like, it can be your vitamins, it can be your hormones. Okay, hormones like your plant hormones. For example, we have, like, like auxins. Okay, so even auxins, they are going to be produced by some of the bacteria, so they are also going to release the auxins, which can be, like, taken up by the plant. Or we can have, like, these plants, they are going to show association with your leaves and all, so they become your endophytes, and then they are going to start giving off all these compounds. So this is going to help the plants to grow much quicker, or they are going to produce some organic acids or organic compounds, whatever that the plant is actually going to require. Okay, so organic compounds, etc. Okay, next thing. Okay, just like this, we are also going to have, like, they also produce sometimes antibiotics, and they are going to protect the plant. So some of them, like I said, they are going to produce certain compounds, and they are going to offer protection to the plant from your predators as well. And finally, guys, when we use these biofertilizers and all, it improves the texture. Okay, it improves texture of the soil. It is also going to improve the water retaining properties. Okay, so the water retaining properties of the soil, and at the same time, this is also going to improve, like, the pH of the soil. Okay, so it is not going to adversely affect, but it is going to improve the pH of the soil, and it's going to try to adjust it so that the plants can grow in a better manner. So these are some of the benefits, okay, of your entire uh, biofertilizers. Okay, so guys, this is what we needed to know with regards to your entire chapter, microbes in human welfare. Everybody all right with this thing, guys? Sure, no issues. Okay, so guys, if anybody has any doubts, quickly ask, or else we'll stop over here for a day, and I will see you in the next class. Till then, take care. Bye bye. Bye everybody. Bye bye. Okay. Yeah. Bye bye. Bye. Yes. Yes. Yes.